So you've seen, when you look at a generic cell in the body, you just assume it's an interphase. If it is in one of these other mitotic phases, it will look different to you. And you should be able to recognize those differences by looking at the nucleus in particular. You want to look at the chromatin and what the chromatin is doing. So in prophase, the chromatin becomes very coiled, very thick and ropey looking. It looks sort of like coiled snakes or earthworms in the nucleus. It's very distinctive. It's kind of like chromatin under a magnifying glass. Despite that coiled look, it's very jumbled. Okay, there's no order or symmetry to it. So that's prophase. During this time, the cell also produces the interpolar microfilaments, those filaments associated with the mitotic spindle apparatus. So the centrioles have to divide during that S phase. They have to migrate to opposite sides of the cell, form their interpolar spindles so that these can become active for movement of chromatin during subsequent stages of mitosis. So prophase. In metaphase, now the chromosomes get ready to divide. So they go from that random coiling or piling of chromatin that we see in prophase. Now they actually take on some degree of organization. And so the first step in this organization is the chromatids line up along what's called the metaphase plate. They line up along the equator of the cell. And so when they do that, they tend to get sort of a symmetrical or quasi-symmetrical appearance. And you can see they're very clearly lined up in an orderly row. That's characteristic of metaphase. Once they do this, now the individual chromatids attach to those microtubules from the centrioles at the kinetochore. Once this attachment is formed, then the kinetochores actually split. And this allows the microtubules to now pull the chromatids apart to opposite ends of the cell. And we see that during anaphase, where the kinetochore or centromere splits, and now the individual chromatids migrate to opposite sides of the cell as a result of the action of microtubules. You remove tubulin subunits, and so that shortens the microtubules and in fact pulls the chromatids to opposite sides of the cell. So anaphase is typified by that kind of bilateral symmetry that you see, kind of like a Rorschach ink blot sort of test. Once the chromatids are, have completed their motion, they get to opposite ends of the cell. Now we are in telophase. So once the chromatids are separated, now the jaw, if you will, of the mitotic spindle apparatus is gone. So now the mitotic spindle disaggregates, it breaks apart, and now the cell begins to divide. The second portion of mitosis begins. Up until now, we've been dealing with nuclear division, or karyokinesis. But now at the end of mitosis, we begin cytokinesis, the actual division of the cytoplasm itself. And you can see that in a plant cell by the formation of this cleavage plate, this cell wall forming between the two units of chromatin. Or in the case of animal cells, you'll see this cleavage form where it pinches in on opposite sides of the cell. So both of these would represent telophase. Once telophase is complete, there are two genetically identical daughter cells which will appear. Now, and then we also have, I guess I didn't show you meiosis. Well, so, and remember the distinct feature between mitosis versus meiosis is that the purpose of meiosis is, instead of producing identical daughter cells, is to produce haploid gametes. And the idea is that you would like each of those gametes to be genetically unique as opposed to genetically identical. Okay, so meiosis produces a variable number, anywhere from 1 to 4 unique haploid gametes, whereas mitosis produces 2 genetically equal daughter cells. Okay, so that's it for cell replication, but as we all know, nothing lasts forever, and so sometimes cells die. And that process of cell death is commonly referred to as apoptosis or apoptosis, depending on who you talk to. There are several sequences of events that you can see during this process, And there are certain words that are used to designate these or that are associated with particular aspects of apoptosis. And so one of them is this nuclear shrinkage. It's very common that the nucleus shrinks and becomes very darkly stained, very darkly basophilic. And so a nucleus that has undergone this process is said to be a pycnotic nucleus. And the process of nuclear shrinkage is known as pycnosis. So out here we've got, just generic, happy-go-lucky living cells, and you can see what their nuclei look like. You can see, dispersed heterochromatin. Sometimes you can see, what's it called? I just lost the word. Nucleus. I'm sorry, that word's gone. Anyway, contrast these nuclei, nucleoli, that's the word. Sometimes you can see nucleolus, sorry. Contrast these nuclei with this nucleus, which is very, comparatively speaking, much smaller. It's at least half the size of these other nuclei, and it has lost all of its nuclear detail. So it has become shrunken and very basophilic. That is a pycnotic nucleus. As this process of degeneration progresses, the cell cytoplasm, as the cell dies, remember, cell pumps in the membrane shut down, so as a result now sodium accumulates inside the cell, that attracts water, the cells swell, they become very brightly eosinophilic, they look basically like large pink balloons. And then the cell starts to fragment and break up. So here you see some of these remnants of these large swollen pink cells, and within those broken up cells you can see nuclear remnants. And this breakup of the nucleus is referred to variously as karyorrhexis or karyolysis. And so this debris can be referred to as nuclear debris. There's different terms you can use, but they're often used, the terms are often used interchangeably.
We would settle whether definitely between karyolysis and karyolysis decisions in, or just are they kind of lumped together?
Do you have to know the difference between karyorrhexis and karyolysis? Essentially, they're the same.
Oh.
Okay, I mean, they're very fine distinctions of a continuing process. But if you can recognize one of them, I'm very happy.
Oh.
That's something that happens as you get older and you compromise many of those ideals you cherished in childhood. You just don't have time. So, blood was the first of the tissues that we looked at. Remember, blood is a cellular, has a cellular and a non-cellular component, so we have cells floating around in liquid plasma. You should be able to identify these cells. One of the major categories of cells, of course, the erythrocytes. Erythrocytes in mammals are enucleate, so they lack nuclei, but sometimes we can still see nuclear remnants. And these nuclear remnants are known as Howell-Jolly bodies, and they're very distinctive. Now, immature red cells that have been released into the peripheral bloodstream may also have some remnants of organelles, like RNA, endoplasmic reticulum, stuff like that. And so, in this case, those cells are known as reticulocytes, and they have a very fine stipple pattern to their cytoplasm. And if you see these reticulocytes in large numbers, that is generally an indicator of an anemia of some type, of blood loss.
Can you go back real quick on that? The reticulocyte? I guess the biconcave disc won't be as nearly as deep, I guess.
If you're looking at cells on a smear, remember in a smear, the cells are going to sit flat on the glass, so you're not going to see them from sideways, so you don't get the biconcave disc. If you're looking at an EM view, where you've got that transmission electron microscopy, now you can see them from the side and get those lovely biconcave discs. Most of what I'll show you will be smeared. Other category of cells are the blood cells are the leukocytes, or white blood cells. Several categories. We've got granulocytes and agranulocytes. Remember, granulocytes, everybody has primary granules. Granulocytes also have secondary granules. So the first of these granulocytes are neutrophils. Neutrophils are also known as polymorphonuclear cells, or PMNs, because they have these very elaborate nuclei. It's a single large nucleus, but it's segmented, so it looks kind of like a sausage with either anywhere from 3 to 5 lobes in it. So if you see a cell that's got granular cytoplasm, and this large, ropey, multi-segmented nucleus, you can be pretty sure it's a neutrophil. Now, in addition to the routine contents of primary granules, neutrophil primary granules also have an additional product called myeloperoxidase, which is an antimicrobial product. Besides the secondary granules, the neutrophils also have a special tertiary granule, which has something else called gelatinase, which is used to help digest its way through collagen fibers and elastin fibers in the extracellular environment. So, neutrophils. And this is just another view, a very nice view of a neutrophil. And it's showing you a bar body, and the degenerate X chromosome that I mentioned earlier. So that came from a female. And here you can, well, never mind, we'll get to that. Next up in the white blood cells, we have basophils. Basophils are very rare, less than 1% of cells in a normal individual. Basophils are called basophils because their specific granules are very basophilic. They stain very dark blue purple. So it may be difficult to distinguish the nucleus because of these basophilic granules in the cytoplasm. If you can see the nucleus is going to be distinctly bilobed, so it will look like it has two halves to it. Basophils tend to be fairly large, like 12, 14 micrometers diameter, something like that. And so when you look at them, what you see is a very large, darkly staining cell with lots of basophilic spots in it. The secondary granules of basophils contain all sorts of nasty things. It includes stuff like histamine, heparin, slow-reacting substance of anaphylaxis, eosinophilic chemotactic factor. So you should be able to rattle off a list of things like that and know what some of those different things do. When basophils leave the circulation, when they enter tissue, they undergo aging, and they are now known as mast cells. Mast cells, to show up well, are generally stained with a special stain, like something like toluene blue or PAS. And when they're stained in that way, these secondary granules become very vivid. Very commonly in tissue sections, mast cells are pointy on one end and rounded on the other, so they look sort of comma-shaped. When you look at an electron micrograph of a mast cell or basophil, these secondary granules show up very, very nicely. So, mast cells. Another very large leukocyte with very prominent secondary granules is the eosinophil. Eosinophils are distinct from basophils because their secondary granules stain bright red. They're not just eosinophilic, they're bright red. So they look kind of like raspberries. And this is a particularly beautiful photomicrograph of the eosinophil. They won't always be quite as nice and eye-poppingly red, but I'll try to find a nice one for you if I ask you this on the exam. Because the specific granules, because the secondary granules are nice and red-pink versus the nucleus, which is purple, the nucleus is generally very visible in eosinophils, and so you should normally see a very distinct bilobe nucleus in eosinophil. The specific granules of eosinophils, so I keep saying specific, the secondary granules of eosinophils will contain another grab bag of products, but in this case, the job of an eosinophil is to counteract the badness of the basophil. So the stuff that you see in eosinophil specific granules includes things like histaminase to break down the histamine produced by basophils. It includes things like eosinophil peroxidase, lysosomal enzyme, major basic protein. Major basic protein will counteract the effects of heparin, and it will also be used as an antimicrobial device, not antimicrobial, but
Parasitic.
sorry? Parasitic, there we go, that's the word. It's used to kill off metazoan parasites, usually worms in the GI tract. So that's it for EOs. Now when we get to the A granulocytes, remember, these are the cells, the leukocytes that lack those secondary granules. So they have only the primary or specific granules in the cytoplasm. So we've got basically two categories, the monocytes and